Hi everyone, and welcome to our video on translocation, the seventh and final video in our transport in plant series. What we're going to be looking at in this one is 3.1.3f, the mechanism of translocation. So we're going to find out what it is and how it actually happens. First thing, as always, let's look at what that word actually means and a few other key terms that we're going to use through the video itself. First thing, translocation. This is our transport of assimilates, and the assimilates are sugars and amino acids throughout the plant. So we describe this transport as being from the source to the sink. Now, where we talk about a source, this is where the assimilates are going to be loaded into the sieve tubes. And where we talk about the sink, this is where the assimilates will be removed from the sieve tubes. And this is all going to be taking place within that specialised tissue of the phloem. First thing then, let's have a look at this process called active loading. So this is where we're going to actually be loading the assimilates into our sieve tubes. So what we're going to be doing is a process that is active, therefore it requires ATP. So active loading isn't just a random name, it is actually an active process, thereby requiring ATP to occur. So what we actually find then is we're going to be taking sucrose and loading it into the sieve tubes. So what we've actually got, if we look at our diagram on the right first of all, we start off with our sieve tube element, which is this part on the left, and then we have our companion cell, the bit that's stuck on the right there. So, first thing that's going to happen is we need to pump the hydrogen ions out of the companion cell. So what we need in order to pump hydrogen ions, as the name pump suggests, this is active, therefore we're going to be using ATP. So ATP will be converted into ADP, an inorganic phosphate, and that means we're then going to have the energy to pump those hydrogen ions out of our companion cell. So what that means is our hydrogen ion concentration within the cell decreases and the hydrogen ion outside the cell increases. So basically what we have is a high concentration of our hydrogen ions outside and a low concentration of them inside. So that means we've now set up a concentration gradient for our hydrogen ions. The next thing that happens is those hydrogen ions are going to undergo this process of facilitated diffusion through a co-transporter protein back into the companion cells. Now, the phrase co-transporter, remember, means that we're going to be transporting two things together. So the other bit we're going to transport in will be sucrose. So what we find is the hydrogen ions and the sucrose are going to then go through those co-transporter proteins into obviously our companion cell at that point. Now, when the sucrose enters the companion cell, that means the sucrose concentration is going to increase, and therefore we've got a higher concentration of sucrose in the companion cell than we do in our sieve tube element. Therefore, it just diffuses into the sieve tube through those little pores, the plasmodesmata. So what we're seeing there, first step, hydrogen ions and sucrose co-transporter proteins allow them to flow back into the companion cell. Then our sucrose is just going to diffuse through the plasmodesmata into the sieve tube itself. Once the sucrose is in that sieve tube, what we're going to find is it just moves along the phloem by mass flow. What we actually describe the inside of the phloem as containing is sap. So the sap itself isn't just sucrose. We also have amino acids and other assimilates present alongside the sucrose. And one key thing to remember about this translocation is that the sap can move either up the plant towards the leaves or down the plant towards the roots, because both of those regions will require those assimilates. In terms of how the plant knows what direction to send things, because it's not obviously consciously thinking, has no brain, then what we actually find is that flow from source to sink is caused by a difference in hydrostatic pressure. Now, what we end up with, if we get a difference in hydrostatic pressure in a tube, we've created a pressure gradient. So in simple terms, first of all, what we're going to find is at the source, water is going to be entering the tube, and that's going to cause an increase in pressure. If we then go to the sink, what we see is a decrease in pressure because water is going to be leaving at that point. So that means that our source will always have a higher pressure than the sink. So the sap is always going to flow from the high hydrostatic pressure, the source, to the low hydrostatic pressure, the sink. Now that we've got that basic understanding of what's happening, let's explain why we get that water movement at the source and the sink in a bit more detail. So, at our sucrose entry point, then what we actually find is the water potential is going to become more negative. So where the sucrose is going from the companion cell into the sieve tube element, that is going to make the water potential more negative. Because we've got a more negative water potential, water will enter by osmosis, because it always obviously goes from a less negative to a more negative water potential. Because water has now moved into that region, we've now increased the hydrostatic pressure. Then go to the other end, the sink. At the sink, we're going to be removing sucrose because that's where we need the sucrose for whatever process we are carrying out. Therefore, as the sucrose is removed, what happens is the water potential becomes less negative, and so the water is going to leave by osmosis. And what we actually find here is it's therefore going to reduce the hydrostatic pressure at the sink. So this is how we've created that hydrostatic pressure gradient, because as the sucrose moves in or out, it changes the water potential. Therefore, water will either enter or leave down to osmosis, and that creates the pressure gradient overall. As always, I do suggest that you subscribe to the channel so you can see when I next upload another video. And of course, head on over to the A-Level Biology website where you can find a range of other resources to help you in your A-Level biology studies.